we have the pleasure to have Professor Paul Watkins. As you can see, he's Howard Ferguson Distinguished Professor at the UNC Institute for Drug Safety Sciences at the University of North Carolina, Chapel Hill. And he will be talking about genetics and amoxiclav DILI risk. Maybe Maribel, you want to say some words before he starts, or?
Well, we just want to emphasize how pleased and honored we are to have this excellent research with such a long-standing career on drug-induced liver injury. It's really a real pleasure to listen to you today, and we are very sure that we are going to learn many things and to enjoy learning. Thank you very much, Paul, for being here with us.
Sure. Thanks, Maribel. So I'll just get started.
Yes, go ahead.
Yes.
So by way of a disclaimer, I'm not a geneticist or a statistician. And I want to acknowledge Pio Nicoletti, who is, and many of you have collaborated with her. And certainly if questions come up that I can't answer, I can arrange for her to be in the loop for that. And so this is a real case report that was included in the Dillon Registry and Biobank. 76-year-old man who developed a respiratory infection and subsequent pneumonia and was treated with amoxiclav and azithromycin dose pack and a Medrol dose pack. And about 3 weeks later, he noted jaundice, was hospitalized, developed pruritus, constipation, fecal impaction, urinary retention, fatigue, 10-pound unintentional weight loss. Bilirubin peaked at 15.5 milligrams per deciliter. His albumin fell all the way down to 2.4 grams per deciliter. His R value was always less than 2, so this was a cholestatic injury. And he did get better. His laboratory values normalized over the next month, but it took 2 more months until he felt well. So this was obviously a very significant adverse health outcome to him. Workup for all their causes was negative, and he never recalled or had any evidence of receiving azithromycin or amoxiclav prior to this event. So the question is, was this amoxiclav or azithromycin? And of course, important thing is to tell him which, if he has to, not avoid both for the rest of his life. And RUCAM score was identical for both, and actually expert opinion at the time this patient was, which is what we use in the Dillon network, 3 separate hepatologists need to agree, or it goes to a committee. We're also on the fence of whether this was amoxiclav or azithromycin. So I'm going to talk about what we all believe, I think, are the sequential steps that lead to idiosyncratic DILI, and then the studies that we've undertaken in collaboration with many on the Zoom call here, and then talk about the role of genetic testing that I think is emerging in terms of DILI, and then some conclusions. So we all know that many, if not most, of what we would call idiosyncratic DILI, and certainly true for the delayed onset DILI events, actually reflect an adaptive immune response. That is a cytotoxic T-cell directed attack on the liver. And there's a lot of evidence for that. This is just looking at with tolvaptan days on since start of treatment. The shaded gray is while the patient was treated. And you can see this is a delayed reaction, not really occurring until 4 months or so after starting the drug, with ALT rising here. But stopping drug, you know, this just grinds on with a mind of its own for weeks, in this case, months, before finally recovering. Then when given the drug, it immediate response, you know, like a memory response. And this is a typical, not always seen, but typical, and supports an adaptive immune response. But particularly, as we all know, it's been as genetics of risk have been looked at, the things that have popped out as the most significant risk factor are specific HLA risk alleles. And really the original pioneering study that was most remarkable was flucloxacillin, European and daily leading with this amazing odds ratio for individuals carrying this common HLA allele 5701. But now with many drugs that are, and this isn't even a complete list now, where the most significant association that can be found are these HLA risk alleles, which code for the proteins that actually hold up the antigen, both in class 1 on all cells and class 2 in antigen presenting cells. And I'll come back to Moxiclav, as having two well-known associated risk allele, one class 1, the O201, and the DRB 1501, the class 2 allele. So what we now believe is going on was really captured in this wonderful figure by Jack Utrick. It actually came out in ChemTox research, so there's actually a reference for it, showing what is believed to be the sequential steps in idiosyncratic DILI, starting with a formation of a neoantigen, and he's sort of showing the standard hapten, reactive metabolite modifying a protein occurring in the hepatocytes themselves here. But just displaying on the surface of cells a new antigen does not initiate a robust response. You need to have some sign that there's disease also associated with it. So for instance, in a vaccine, COVID vaccine includes what are called adjuvants, which are really to create inflammation at the site of injection. The reason your arm is sore is just simply giving a modified protein is not good enough. And so the idea is that the drug, in addition to making a neoantigen, is causing inflammation in the liver. And it does not appear that requires death of liver cells, but stress of liver cells with release of what are called danger signals or damage-associated molecular patterns to stimulate immune cell recruitment, activation of macrophages, to create this, in a way, to have the adaptive immune system believe the liver is virally infected. There's actually, a disease there, in addition to the neoantigen to prompt a cytotoxic T cell attack. And then the idea is that you probably need the same neoantigens that are being made and put on the surface liver cell also need to get to professional antigen presenting cells, again, to have a robust response. And the leading thought now is that may be occurring in exosomes that then are released and taken up by the professional antigen presenting cell. And then that professional antigen presenting cell will put that antigen on class 2 HLA molecule along with co-stimulatory signals to find and bind to T cells with the appropriate T cell receptor, then leading to activation and expansion of those T cells, which can then hone back through circulation to the liver to react with the hepatocytes that have the same antigen on its surface and through various means actually kill the cell. And that's maybe the first sign of liver injury, actually. And often that leads to nothing. It stops because of immune tolerance. But in the case of clinically important DILI, it progresses on to clinically significant in the absence of immune tolerance. And so I think Jack did a real nice job of sort of summarizing at least my current thinking of the fact of what's going on here. And so studies in amoxiclav are supporting this general idea. And of course, the first, I think it was Mirabel was lead author in this gastroenterology paper that looked at about 200 cases of augmentin DILI and just 532 population controls and did genome-wide association. As we all know, the way you sort of visualize that is what's called a Manhattan plot, where you have the chromosomes on the x-axis and then the significance of the difference in incidence of specific SNPs in those cases versus controls. And because you have many many, many SNPs, currently in the megastrip 6 million, the level of significance to correct for multiple comparisons has to be a p-value with, 7 or 8 zeros ahead of it. And when this was done, clearly there are multiple SNPs in the short arm of chromosome 6 that were indicating a strong difference between cases and controls. And that turned out to be related to a class 2 HLA molecule, DRB1501, which is presumably what is on the professional antigen presenting cell, putting up that antigen to activate the appropriate T cells that have the right T cell receptor, but also a class 1 HLA molecule, O201, which is presumably the main HLA molecule holding up that antigen on the surface of the hepatocytes. And a nice thing in that paper is they showed the risk for DILI was about increased twofold if you had the HLA class 2 HLA allele, and about twofold if you had the class 1 HLA allele. If you had both, it was about eightfold. So there was actually this, what they call epistasis, a synergy, presumably because of, this mechanistic link, between the two here. So the next question is, we know from a variety of studies that probably about 40% of susceptibility to DILI and augmentin DILI also is genetic, and about 60% is probably non-genetic factors. And these two account for less than 10% of that variability. So there's a lot of genetic variability we know that is left, that's underlying susceptibility, and how can we get at it? So this is where this manuscript, again, you can see collaboration, latest in a series with European investigators, some on the line here, I know, particularly pro-EuroDILI, you guys. And this came out a couple of months ago as an EPUB, and actually just yesterday came out as a actual publication. And I'm going to be focusing on what's in this for the rest of this talk. So what this allowed us to do for the first time was have enough cases of amoxicillin clavulanic DILI to create a discovery set and also a validation cohort. So we had 444 in our discovery cohort, and then we could test hypotheses in the validation cohort. And that really allows us to steer away from GWAS, because if you have an association that's strong, a good biological plausibility, and you can validate an entirely different cohort, the validation cohort, especially with a whole different set of controls, which we had to put together, which Paula Nicoletti, did, and was a major effort. You can begin to get very credible evidence for associations that aren't GWAS significant. And if you just want to look at this quickly, they're very comparable in the phenotypic characteristics and also the biochemical characteristics across the discovery and validation cohort. So the first thing we did, both in the discovery cohort and when we put them both together, was say, if we can condition on the known HLA risk alleles, the class 1 and class 2, is anything else GWAS significant? And that's shown here. And the answer is no. There's nothing else, which is too bad, because this is, without any hypotheses, that's how we've discovered things. But there was nothing that was GWAS significant. But we felt pretty, because HLA has been such a recurrent theme, we thought, as most people do, you don't need GWAS significant. Let's just look among the HLA risk alleles and look for statistical significance in that group. And it turned out in this manuscript is reported another, it's actually a rare allele. It's only about 1% carried by the population. This a class 1 B1518 that was significant in terms of just HLA risk alleles, if you use that as the multiple correction, and validated in the validation cohort. So we came up with a second class 1 HLA risk allele. But then we also felt comfortable putting in to this model this non-synonymous variant in PTPN22, even though it didn't actually validate in our validation cohort. And I'll show you that later. And the reason we were confident to believe this is a key susceptibility factor in augmentin DILI was a paper that came out in 2019. Liz Cerulli was the lead author, again, a collaboration with the Europeans, that looked among all the cases we had at that time, 2,048 DILI cases, but conditioned on the 4 known HLA risk alleles at that time. And when we did that, lo and behold, in chromosome 1, there were significant SNPs, which turn out to tag a non-synonymous variant in this gene PTPN22. We validated that actually with help of Einar Bjornsson and Iceland, the DECODE registry that they have. So we were very confident. And furthermore, when you looked at the actual drugs that were contributing to this association, GWAS significant across all drugs, Augmentin was at the head of the pack. Not GWAS significant, but very significant. Even when you took it out, you get significant associations. By the way, if anyone wants to interrupt me and ask a question at any time, that's fine too. I am planning to leave some time at the end. Interesting, flucloxacillin was not on this list. This is just those that had a p-value less than 0.1. So we were very confident, even though we didn't validate it, to include this as a reasonable risk factor for augmentin DILI. But then we did something which is called transcriptome-wide association study. And this is where you take advantage of publicly available databases available through GTEx, where they've done GWAS on patients that they also have tissue from, and so liver or blood or other tissues. And they then just look to see in the GWAS SNPs, are there associated differences between having the SNP or not having the SNP and expression of specific messenger RNAs. And so if you can go there, and they have done this for about 200 liver samples. And so you can convert the GWAS to the predicted expression level of specific mRNAs that are in the liver. And this is something being pretty widely done now, look at different tissues. And really the reason to do this is 3 I could think of. One is it reduces the number of variables. So if you're talking about the MegaX chip of 6 million SNPs, you're going to convert that to the expression of about 20,000 genes. And actually, if you just look at liver, it's even less. There varies depending. It's about 10,000 in liver. So in doing that, you're lowering the threshold for significance substantially, orders of magnitude, when you look for differences. And again, there's some statistical concerns, but if you have a validation cohort, you can build a convincing argument. Also, and Ann Daly was, I think, the person I know that's most involved in this when the Severe Adverse Event Consortium actually paid for whole exome sequencing on about 200 AC DILI cases. Not a lot came out of that. There was never a publication, for instance. So it certainly wasn't supportive that non-synonymous changes in the exome are going to be major contributors to risk. And then lastly, there is evidence that genetic susceptibility to DILI may largely relate to regulatory variants, actually, not exonic. And this is a paper that came out of Takabe's lab. Some of us can help contribute to it. It's a very difficult to follow Nature Genetics or Nature Medicine, Nature Medicine, there it is, manuscript. But what was done there is a true polygenic risk score, actually taking the Cerulli et al. Data on cases, and actually just looking at cases of cholestatic and mixed DILI, and then doing what's called create a polygenic risk score. And what you do there is you take the GWAS, and then you basically lower the threshold sequentially, of course, getting hundreds and then thousands of SNPs that have a different frequency between cases and controls. And there are various software things to do this. And so he he did some funny tricks. He looked at only genes that are expressed in the liver and then picked a cutoff that maximized the difference between cases and controls. But then he looked to see where are these SNPs that are actually part of this large cluster that seemed to differentiate cholestatic mixed cases from controls. And what he showed was this diagram, which the way you read this is sort of the further out from the center is the higher the percentage of SNPs that are in the various areas. And there were some exonic SNPs shown here, but the major contribution in this polygenic risk score were actually in the 5' untranslated region, and concluding that at least with cholestatic mixed DILI and these, included augmentin, that the major contributor were actually not exonic, but presumably regulatory variants. There you go. So we did that. And so we took the 444 discovery cases and we converted the GWAS into the predicted expression, comparing cases versus controls. Again, our very large number of patients of people of European ancestry. And then what you do is you've lowered a couple orders of magnitude the level of significance. And lo and behold, there was one gene, one mRNA predicted to be expressed significantly differently in cases and controls. And this is this gene called ERAP2. Actually, it's a common, this variant is common, carried, by about 50% of the population. And it was actually lower expression that was associated with risk rather than higher expression of ERAP2. And this is, very interesting stuff. There are two enzymes. There's ERAP2, which is the one I'm talking about, and also ERAP1. And these are little molecular scissors that sit inside the endoplasmic reticulum. And as proteins, cellular proteins, are degraded in the proteasome to peptides, the actual fragments that are generated and go through this thing, tap into the endoplasmic reticulum, are too big to fit into the peptide binding site of class 1 MHC molecules. And ERAP2 and ERAP1 actually snip them down into size. And it's very interesting. Sometimes ERAP2 and ERAP1 will snip too small so that they don't even fit in there anymore. And they have sort of different substrate bindings and different links that they make and everything. But it's all a way of controlling what antigens end up in the class 1 MHC molecule as it's synthesized in the endoplasmic reticulum before it then traverses to be presented on the surface of the cell. So this is all part of determining self from non-self, and only about 20% of antigens that are generated actually end up on the surface of cells, in this case hepatocytes. And this would then presumably help control what antigens are projecting out there for interacting with T cells receptors on T cells. Now, it turns out there's a great rising interest in ERAP2. So if you go to PubMed and you just search ERAP2, what you find is almost nothing till a little over a decade ago, and then a steady increase in number of publications, obviously just beginning of 2023. And it turns out for ERAP2, there are two major haplotypes. There's HAP-A, which is associated with autoimmune diseases, actually birdshot retinopathy, ankylosing spondylitis, and some psoriatic diseases, expression of HAP-A. And then there's HAP-B, which is actually what our SNP is tagging. And it is a truncated and rapidly degraded mRNA and has not been associated with autoimmune diseases. And so we know that what we found, at least the conclusion is that carriage of HAP-B increases your susceptibility to AC DILI, amoxiclav DILI. But In the case of birdshot retinopathy, it's actually the opposite. It's HAP-A that has more expression, higher expression level, more enzyme activity. That's actually the risk factor for birdshot retinopathy. And there is an associated strong HLA risk factor called HLA-29. And actually, it's been shown in cellular experiments that HAP-A trims peptides and has the profile that is unique, aspects that are unique to binding in the peptide binding site of HLA-29. So in the case of birdshot retinopathy, the actual link between the risk allele, which is HAP-A, and the presentation on the antigen has been linked even in terms of the profile of peptides that are generated. This is an amazing paper to me. This came out just a couple of months ago in Nature, and it was talking about the Black Death, the bubonic plague, Yersinia pestis, and actually, I'd forgotten that during the 14th century, actually the population in Europe halved over several years because of this plague, and they incidentally have discovered mass burial sites of patients, I guess when they're doing construction on new homes and things, that are before, during, and after the pandemic that hit, the epidemic, the Black Plague, both in England, which served as a discovery cohort, and in the Netherlands, which was the validation cohort. And they then looked to see common alleles that are known to influence immune function, with the idea that those that survived may have certain alleles in the in immunity that help protect them. And the one allele that came through in this study was HAP-A, or ERAP2, just coincidentally. It turns out that after the bubonic plague, the European population was significantly enriched for this HAP-A haplotype of ERAP2, and so the having that actually was a survival help. But it also then, the argument in here is predisposed European populations to autoimmune diseases. Anyway, it's a fascinating paper, and I recommend you take a look at it. And they also, by the way, showed in cellular systems that HAP-A macrophages with HAP-A were better able to respond and react to the Yersinia pestis bacterium. So what they're saying is that, as a result of the bubonic plague, the European populations were enriched with this HAP-A that exists to this date, and that's the reason has contributed to increased susceptibility to autoimmune diseases. Now, when I saw this, I got excited because I said, actually, am I going to show this? Maybe I'd show it later. Sorry. So the other thing is there is a other ERAP enzyme, which is the ERAP1, as I showed you, and that actually has 10 haplotypes. And the haplotype 2 has been associated with high activity of ERAP2, and 8 and 10 have been associated with low activity. And we did look in our AC DILI cases at, where did those stand? And the HAP2 was enriched, relatively enriched in AC DILI cases versus controls, and the low activity was relatively unenriched. And again, I won't go into this in much detail, but this supports the idea that the risk for augmentin DILI is actually linked to peptide trimming by the ERAP1, the haplotype 2, both the fact that the HAP-B means you have very little of haplotype B, and the fact you have enriched for HAP2 means you have more of ERAP1. So it's consistent with the idea that's out of birdshot retinopathy that the whatever this antigen is, and that's a fascinating story in itself, is being preferentially presented, trimmed and presented to A0201, and maybe this more minor, less frequent HLA allele to be presented on the surface, and that this is a similar mechanism. So the take-home point is the people of European ascent, it appears that the Black Death, bubonic plague, resulted in increased susceptibility to autoimmune diseases, but less susceptibility to AC Dili. And when I saw this, I thought this is great. Apparently the plague wasn't in Africa, and there are well-known differences in susceptibility to, I'd say well-known, but there's evidence that susceptibility to AC Dili is lower, is different in African Americans than Caucasians. Unfortunately, it goes the wrong way. And this is Naga Chalasani's paper that showed, although the classes, the rank order of classes of drugs causing DILI is very similar in African Americans and Caucasians, amoxiclav is the leading by far cause of DILI among Caucasians, whereas it's number 4 among African Americans. So unfortunately, it doesn't explain that. It's actually going the opposite direction. All right. So how about ERAP2 HAP-B is a risk factor for AC DILI? How about other drugs? And so we looked at all drugs where we had enough cases, and the answer is no. It's not a risk factor, even for flucloxacillin with a very large sample size. But interestingly, it did come out as a risk factor among our 20 cases of DILI that had been attributed to amoxicillin alone. That is amoxicillin without clavulanate, not amoxiclav. And we wondered why that was. So could it be that we got it wrong, that the patient said he was taking amoxicillin, but in fact it was amoxicillin clavulanate? I don't think that's the answer, and I'll show you later evidence that supports that. The clinical presentation of these 20 cases is not identical. It's not characteristic for augmentin DILI in a number of ways. But also Dean Nesbitt, showed that in patients who had recovered from amoxiclav DILI, he was able to find clones of T cells that reacted with amoxicillin protein adducts. So it may be that, although we traditionally have thought of clavulanate as the bad actor in the mix, that actually in some cases amoxicillin may be in fact contributing to the risk. All right, so where are we now? So we got the class 2 HLA, we got the class 1, 2 class 1 HLAs, we got PTPN22, and now we've got ERAP2 and maybe ERAP1. I didn't make a big deal out of that. They're close on the chromosome, so there's some linkage. So we can't be confident that the associations with the HAP2 of ERAP1, are yet there. But again, pointing out that ERAP2 is in there. So we've got 5 separate risk factors for AC DILI. So one of the things that we then looked at is something that's not been very talked about in terms of genetic risk for DILI, although maybe it's obvious, and that's the importance of zygosity. So these are our 5 markers that we're talking about. You can see if for the class 1, if you're homozygous, you get roughly twice the risk of being heterozygous. If you're with the class 2, ditto. In fact, maybe even more. Might even be more risk being homozygous. There weren't any homozygotes for the, I think, or maybe 1, so we couldn't really say anything about this rare allele that validated class 1 allele. Interestingly, with ERAP2, it was only the homozygotes that had increased susceptibility. The heterozygotes didn't. And we were actually able to go to a large database where GWAS has been done in over 1,100 livers. That's now actually, the database is publicly available, and looked, and actually the level of mRNA for ERAP2 is not lower in the heterozygotes. It's only when you get the homozygotes. So there's some sort of compensatory mechanism for the heterozygotes that maintains expression, at least at the at the RNA level, mRNA level for ERAP2, but also doesn't confer risk. The heterozygote doesn't appear to have any risk. So that's just an interesting observation. And then for PTPN22, again, about doubling of risk, whether you're homozygous or heterozygous. And there was, even though we couldn't validate the association, the validation cohort, there was a trend towards this homozygous being more, being, higher risk than heterozygous. So we were then able to calculate what's called a genetic risk score, which is just taking from sort of regression analysis, the allele dosage, 0 heterozygous, homozygous for the class 1, 2 class 1 HLA, class 2, the homozygous ERAP2 status, and then the allele dosage of PTPN22. And actually in the paper give examples of how if you find an individual, spelled wrong, carrying one HLA class 1, this one, how you would actually come up with a score in this case. But in order to apply it to risk of an individual of developing AC-DILI, we had to have an estimate for what is the average person's risk of developing AC-DILI. And fortunately, thanks to Einar Bjornsson and the wonderful electronic medical record system in Iceland, we were able to come up with an estimate. And this is a table from this manuscript. And so you see in 35,000 patients that were treated with amoxiclav, 15 cases of DILI were observed, which is about a 1 in 2,350 incidence, and sort of an average European. And so if we use that, we can then create quantiles of risk based on this genetic risk score, where roughly 1 in 23,000 is an average risk score calculated, because these are common variants of between 1 and 1.5. But depending on what your score is, that might increase to less than 1 in 5,407, or as frequent as 1 in 100, roughly. And we had looked at both the discovery cohort and validation cohort and got similar results. So we then looked to say, okay, how specific is this genetic risk score for amoxiclav versus DILI due to other drugs? So we looked at other drugs, and what you can see is obviously amoxiclav is highly specific. We developed it for that, but there really is little sign that it's useful in flucloxacin. In fact, it's in the other direction. The odds ratio is actually less than 1. I won't go into specific how this was calculated. But then amoxicillin alone, also, if anything, the direction is in the opposite direction. So I think the association we found with, and in spite of the association with ERAP2 and amoxicillin DILI, there was no association with the other variables, and it probably is a real association with amoxicillin and not just mislabeling or mislabeling what drug they were taking. So how do we actually do causality assessment? Now, the big difference between the RuCam, at least the strict RuCam, and what we call expert opinion is that RuCam doesn't and can't take into account the fact that drugs that cause DILI tend to do so in a characteristic fashion. Dilantin tends to have hypersensitivity, rash tends to occur in the first month, tends to be hepatocellular. Augmentin tends to be mixed hepatocellular, tends to occur in older men, et cetera. And of course, the LiverTox website, which is the really genius and lifelong effort now of Jay Hoofnagel, is really the place to go to find out what these signatures are. And I know people, like Raul, who use RuCam, will also consider, this kind of RuCam score of 8, but actually it doesn't really fit what's been described, you know, sort of putting that into the equation. Well, how does our genetic risk score stand up to the characteristic presentation and risk factors for Augmentin-Dilley? And we go over this in the manuscript. First of all, Augmentin-Dilley tends to be more common in older men. It has a characteristic latency, you know, about 3 weeks after starting a course, you see the event. That's different than other DILIs or some other DILIs. And then it tends to be cholestatic or mixed versus hepatocellular. So that's sort of the both risk factors and clinical presentation. And we use that, and I think everybody uses that when they're assigning causality to a drug. Is it the right clinical setting, and is it the right biochemical type, and does it have the right latency? Is it consistent? And so when put into a multiple regression, sure enough, in our cases, age, sex, latency, you know, versus other drugs, being cholestatic or mixed versus hepatocellular, in the discovery cohort, less so in the validation cohort, so just concentrate over here, were significant variables. But even in the model, putting all those in together, the genetic risk score was significant. In fact, it had the highest odds ratio and the highest p-value. So this genetic risk score was adding to the current way we do causality assessment, even for a drug like Augmentin-Dilley, where there is a well-known sort of characteristic presentation. So going back to the case, let's see where we are. This looks good. 76-year-old man, again, he was getting both Augmentin and zithromycin, had the same RUCAM score, same expert opinion that couldn't distinguish between which ones. And actually, his genetic risk score, when you calculate it out, is 2.7. Where does that sit? Well, in our table I showed you before, 2.7 is right in here. So his risk of developing DILI is about 1 in 600 here, 1 in 400 in the validation cohort. Significantly greater, obviously, than the 1 in 2,300, which was the sort of the baseline prediction. And and in reassessing this case, we all felt pretty comfortable saying that this was amoxiclav DILI and not azithromycin. And you could argue maybe we should have thought that from the beginning, just based on the incidence of relative incidence of DILI from these two agents. So we know this accounts for actually about 13% of risk in the regression analysis. And so we know we're still missing about 2/3 of the genetic risk of AC DILI. And how are we going to find it? So I'm ending up here. Well, there's a guy named Johnny Sexton working with Bob Fontana that's actually making induced pluripotent stem cell derived organoids from our Dillon patients that have had amoxiclav DILI. It's a bold move because unlike, making organoids from iPS cells for conditions where the specific susceptibility factors, alpha-1 antitrypsin, it's not easy to try to assure yourself that you've got mature organoids that are expressing the right combination of things when you don't know what those combination of things are. So for instance, ADME genes, CYPs and transporters haven't come up in any of these analyses. And ERAP2 is probably occurring at the level of the hepatocyte, but not necessarily so. So we've yet to find really any susceptibility factors that are going to be for sure at the level of these organoids. On the other hand, we believe they're there. They have to be. So that's a way to sort of test hypotheses, compare these cells to other cells, treat them with Augmentin. The other thing we've been exploring is, it's possible in GTEx with TWAS to look at to predict the liver expression of cells. But now databases are coming up because of single cell RNA and proteomic profiling, where you can begin to look at genes that are only expressed in subcell types in the liver. So, for instance, there's a particular gene, mRNA is shown as the brown dots, and this is a gene that's only expressed in hepatocytes, not in cholangiocytes, not in macrophages, not in endothelial cells. And you can now do that TWAS, for instance, and say, let's only consider variants that are in or near or associated with expression of hepatocyte-specific genes or cholangiocyte-specific genes. And this, you get into some issues in terms of statistical significance. But again, with the validation cohort, I think this will be an exciting way to go. We're also hopeful for whole genome sequencing, but can't find anybody willing to pay for it. So my final take-home point is that I think combining genetic testing with clinical risk factors should improve causality assessment, although we're at an early stage here and focus just on amoxiclav DILI. But I believe we'll also progress on to have a role in actual risk management. So that's my final slide. This is our Dhillon group a few years ago, pre-COVID. Of course, Jay Hoofnagel here, Bob Fontana, who many of you know. Paula Nicoletti, as I said, is our heavy lifter in terms of genetics. And then to list sort of some key people that have been absolutely essential, and some of whom are on the phone, but really all those that are involved in the Dhillon networks over in Europe. We're extremely appreciative and excited about going forward. We believe our Dhillon network will be funded until 2028. That's what we're told. And we're looking forward to continued very productive collaborations. And with that, I'll open it up for questions. I guess I'll leave it on this.
Thank you, Paul, for this extraordinary talk. Do you have any questions among the audience? Yes, Anne, please go ahead. Anne
Okay. Well, first of all, thank you, Paul, for a great talk. Now, I have a general comment about what's been seen for co-amoxiclav and amoxicillin for that matter, and why we don't see it for other types of DILI. Now, I realize that we have access to more cases for the co-amoxiclav than anything else. But for example, it doesn't look like it's coming up in the same way for flucloxacillin. So it was interesting. That paper you talked about, the Black Death paper, is of course very interesting. And it raised another question with me, and that is whether some of the risk factors that are coming up for the DILI are actually due to the fact that of the type of infection that co-amoxiclav is used to treat. Because most of the other causes of DILI are used for different types of problems, whether they're bacterial infections or something entirely different. So that's my slight problem with some of this, whether the ERAP2 especially, and also the PTPN22 are genuinely DILI genes, or whether they have something to do with the drug, the reason for prescribing the drug in the first place, maybe as well, I don't know. But that's just one observation. The second observation
that's a very good point.
When you look at polygenic risk scores, we only know of 2 polygenic risk scores in DILI. And the problem with the 2 of them is it's either a very small polygenic risk score as being described today, or the Takanori one, the enormous one, which involves, I think, about 25,000 SNPs. Whereas other polygenic risk scores for things like cardiovascular disease and breast cancer are for a couple of hundred SNPs. So are we going to get to the situation with some of the other diseases where you could genotype for a couple of hundred SNPs and get something?
Yeah, I think this is a point that was mentioned in the paper. is that the fact we've already got 13% of the predicted total genetic risk is with just 5 variants, is very encouraging that there may be a limited number. I'm not sure that's true, but that's what Paula, I know, Nicoletti thought. And actually 13% of genetic risk is a huge amount. And if you started looking at schizophrenia and bipolar disorder, diabetes, and other diseases. So I think the hope is that there, with all drugs, there will be, a good deal of the total genetic risk explained by a few variants that you could, have a SNP chip that would include for the major drugs. Probably first in drug development, where if you have a clinical trial going on with 1,000 people, companies very much want to have some evidence it wasn't their drug. And so therefore to have some sort of chip for susceptibilities to the most commonly used drugs that might confuse interpretation. Is it the study drug like Augmentin? But I think that's unknown. What Paul has been doing, the problem with a true genetic risk score where you keep lowering the p-value and seeing what it does is you need to have validation cohorts. And that's what Takaba, used that TAC 875 clinical trial data for. And it's very hard to convincingly do that with small numbers of, some small sample size. So what she's been doing is looking at large sample sizes that are available and creating polygenic risk scores, say for, various liver disease, autoimmune hepatitis, and then seeing if that separates cases from controls in DILI. So going the other way around, looking at, hoping to see similarities and sort of be able to sort out which genes are involved. But anyway, I'm sort of rattling. I mean, the hope is that there will not be, 20,000 genes that have to be considered in order to fully define the genetic risk of Augmentin or other DILI cases.
Yeah, I agree with that. And the fact that we've got an HLA association in most of these anyway makes it complicated because it's a little different to some of the other types of polygenic risk scores that have been described where there's no HLA association and where the risks are just a lot of combined small ones.
Yeah. I'm intrigued about the idea that you're saying maybe it's the indication for Augmentin. One of the things that's remarkable, in the United States, there are databases at how many people each year are treated with Augmentin or amoxicillin, and it's in the, tens of millions. It's cheap. It's extremely common. I'm told it's a rare adult in America that hasn't received a course of Augmentin, just in the course of being a kid, ear infections, etc. So I think that may argue a little bit against your point and justify population controls versus drug-treated controls. But what do you think?
Yeah, well, I had a bit of an argument about this with Paula because in the UK, Augmentin is very rarely prescribed in primary care.
Right.
It's very much a second line drug compared with amoxicillin. I mean, amoxicillin is really, really widely used, but Augmentin tends to be reserved for more serious infections in the UK. I don't know if that's the same throughout Europe, though, and probably some of our Spanish colleagues would be better placed to comment on that one.
Raul, you want to comment on that?
No. My question was another question. So.
Okay.
Javier, but I can tell you, and I can guarantee that I agree on the point made by Paul. In Spain, is widely prescribed amoxicillin, and we have always considered that as being so widely prescribed, the control population could be an open population. It does not have to be much to the drug, to amoxicillin clavulanate, because being tolerant to amoxicillin clavulanate because it's so widely prescribed that you can consider all population as having been exposed at any time in their life to amoxicillin clavulanate, at least in Spain.
It is an intriguing idea. I mean, Yersinia pestis is a bacteria, I believe.
Yeah.
So susceptibility to bacterial infection. It's interesting. I'm glad, Anne, you didn't bring that up before we submitted the manuscript. I would feel I had to talk about that, or we'd have to talk about that. But it is an interesting angle because this is very specific for Augmentin. And very interesting. So I think, Raul, you said you had a comment or a question?
He has a question. Yeah.
Well, first of all, thank you very much and congratulations for this masterful talk. It was fantastic. And my question is about the translation of this polygenic risk score, for example, amoxicillin clavulanate clinical practice for causality assessment. You mentioned that for causality assessment is going to be useful, but how do you foresee that it can be implemented in clinical practice? For example, in expert opinion, for example, as an addition to the new RECAN, in the new domains, in the new domain of RECAN, and only, for example, like in the case you mentioned, you show us to us is when there is another antibiotic which is also incriminated in DILI reaction, or how do you foresee that?
Yeah, well, to get the genotyping of these 5 variants is not commercially available to my knowledge. HLA, now even HLA sequencing is routinely done in major medical centers that are involved in transplantation. Mm-hmm. So the plan now, and I think Bob Fontana and Skip Hayashi, I know they're working on trying to figure out how to put HLA associations into the RECAM as part of the, does it get a point or not kind of thing. I think this with the the genetic risk score for Augmentin, the first place that will be applied, I think, will be in clinical drug development, where a drug company wants to try to prove it wasn't their drug in the clinical trial, and they have the resources to do the genotyping as part of the weight of evidence that it was not their drug or that it was their drug. And I think Skip Hayashi, at the FDA, who leads, will actually suggest that, if they're saying, well, the patient took Augmentins 3 weeks before the seminar, say, well, have you done the genetic risk score? And I think they'll do it. And hopefully this is just the beginning, as we fill in the rest, it'll become, but I think anyway, and we will use it in Dylan for sure. In other words, we're doing GWAS genotyping on everybody, and we will have a genetic risk score for any case where Augmentin or amoxiclav is considered a potential culprit. But I think it will be in clinical drug development, and then hopefully eventually into clinical practice. But the problem are these very rare events, and now, fortunately, as you know, the associations for many drugs like lamiracoxib were actually, with HLA, were discovered in the larger percent of population with largely asymptomatic ALT elevations. So I think this may be paving the way better for using a genetic risk score, using that larger denominator that may be feasible in a clinical trial setting in a phase 3 database to be able to find these associations, to actually link it to approval of the drug to have genotyping. But drug companies are very resistant to that, and but I think that's going to happen ultimately. We will go.
Thank you.
Any other question?
Well, I would like to just following your thoughts and the reply to Raul, Paul, congratulations, first of all, to this wonderful talk. Well, I remember that in the gastro paper with amoxiclavulanate, North Europeans HLA-A allele was different to South Europeans. The validation cohort, I just wanted to ask you, because in the validation cohort, it was in the same, it had the same trend, but sometimes you could not find the same associations. Do you think, what do you think, or what could you envisage are the main differences? I mean, there might be other genetics, other differences in other, because you have tried this in European population, but mainly UK and Iceland. Do you think that, for example, in Africa or in other populations, you might expect the same associations, the same risk alleles?
Yeah, well, we did look in the manuscript in the few African American and Hispanic cases we have, and there was no suggestion that the genetic risk score that we developed in people of European descent would have any use in those populations.
Right.
And I think, that's a major problem, particularly when you're dealing with HLA alleles. Different populations, different part of the world have had different selection pressures and have different profiles. And, that's what this guy, David Ostroff and others, are trying to group HLA risk alleles, not based on their sequence and, what we call them, but what similarities in their peptide binding site, to be able to look across different populations to say, yeah, it's a different allele, but it has very similar peptide antigen binding properties.
Yeah.
And let's look there, to test hypotheses based on these, what I call HLA supertypes. But the differences across populations is a huge challenge. And it's certainly a challenge for drug companies to get a drug approved. If they only have, Caucasian, European descent patients in their study and they find a genetic risk factor, how are they going to make sure that only those people get the drug? Is that ethical if only those people get the drug? Yeah. So it's more, I'm just saying I'm agreeing with you. It's a challenge. Yeah. The only thing that didn't, by the way, validate of the 5 was the PTPN22. And what delayed the manuscript for a long time is Paula was convinced that was because we didn't have the right second comparator group.
Yes.
And trying, because it was heavily Ashkenazi Jews for a while and everything, and tried everything she could to match up, in a PCA analysis, the controls in the population. But it turned out we never were able to improve the lack of validation.
Yeah. Yes. And also, I would like to ask you: do you think it will be? We are at a stage in which we could try to come up with a HLA map analysis, like a platform for testing HLA that have been identified with associated to some other drugs that for daily risk or serious cutaneous adverse drug reactions. In the specific case of these risk alleles that has been for amoxiclavulanate, do you think is feasible, is easy to perform in a clinical laboratory?
So in other words, some sort of chip that would give you rapid detection of the HLA risk alleles that have been associated with different drugs and DILI so far as a commercially viable product that would be widely used by LabCorp and so I think that's an interesting idea. I'm not the right person to address that, just the technology aspect. And how much of a problem that would solve. But I like the idea, and over time it builds and maybe starts in clinical drug development as a tool they use to help causality assessment. But
it could be, no?
It could be.
It could be.
I mean, it's true. That's another thing I should say. If you look at the contribution to total prediction of the 5 risk factors, the vast majority is in the 2 we knew all along. It's the class 1, class 2 HLA. We're sort of bumping it up incrementally. But, as the GWAS showed that, those are the major risk factors. And those are the HLA risk factors is what they're talking to try to build into the RECAM instrument. So having a commercially accessible way to get that information, makes sense to me.
Yeah. Could be a global effort because some HLA alleles has been identified for Asiatic population, particularly others European, maybe in Africans. I'm thinking of neviparin. Well, I think it will be worthy the effort, no? Yes, now that we have the knowledge and to translate that knowledge into the clinical practice and to be available to patients, because in some cases it gave us the proof or give us more certainty to ascribe a drug to the daily patient, to ascribe that particular drug to the clinical picture that the patient is presenting.
Yeah. Good points. I think Andreas.
Yes,
has
Andreas
his hand up.
Benedict. Yeah. Andreas, go ahead, please.
Hello, I hope you can hear me. Thank you for this excellent talk and congratulations to this great data. I have one question regarding amoxicillin clavulanic acid. Since this is a drug combination of 2 drugs, and I'm quite intrigued what you said earlier, that there were some patients responding only to amoxicillin. So can your data or your patients be subdivided in perhaps 3 populations, the ones that respond to amoxicillin, clavulanic acid, and the combination? Just an idea?
Well, there's no indication for giving clavulanic acid by itself that I'm aware of. So we either have the combination of the 2 or just amoxicillin. And there's no question that amoxicillin is far safer for the liver than the combination. So I think there are 2 groups, right? There's the combination and there's the amoxicillin alone. And unfortunately, we don't have a lot of cases of amoxicillin alone because it is so safe for the liver. But,
okay, but in the AC group, are there some patients that look like the amoxicillin patients? Because from my experience in Munich, these patients have some kind of a different reaction than the classic amoxicillin clavulan.
That's right. There are differences, and we included in the supplement a comparison in our case of amoxicillin that's been updated because Herb Bronkowski had an earlier paper versus classic combination treatment, and they are different. I think latency was a little different. They weren't clinically the same. And I didn't mention that, with all these risk factors, if you look at them individually or together, it doesn't seem to differentiate or be associated with different phenotypes in terms of latency, hepatocellular, cholestatic, mixed. At least nothing very convincing has come out of that. I'm not answering your question, though. But if you have ideas of how to explore this further, I'm all ears with, the amoxicillin versus a combination treatment.
Yeah, thank you very much. I think I'll come back to you. I have some data left from Munich. So
Good. Yeah. This is good. This is good.
thank you.
Excellent. So, do you have any other question from the audience? I don't see any other one. Well, I think that's everything for today. So thank you very much, Paul, for this extraordinary talk. And again, we want to thank you for it's been a pleasure to listen to you, and thank you once again. And Maribel, you want to just close the session?
No, of course, very pleased, Paul, to be able to have you with us in this Pro-Euro-Dili network that is part of ECOS Action. More than 28 countries, 28 countries.
28, yeah. 28.
Is across Europe, and also those that have been what they say target countries, inclusive target countries, recently incorporated into Europe. We have done intense networking, and as part of this networking activity came the idea of having these young conferences series, particularly for young people who did short-term scientific missions to present their results and experiences, and to share that with everybody. But now that we are reaching the end of this ECOS Action, we wanted to have great leaders sharing the knowledge and talking to us. So thank you, Paul, and congratulations for the great work that you are doing, trying to advance knowledge, understanding, translating that into service for pharmaceutical industry, drug discovery, clinical drug discovery, inspiring all of us, which is fantastic. Thank you so much, Paul.
Well, thank you. And I know Dylan and me are really counting on continued great collaborations going forward. It's really an exciting time for Dili.
Yes, that is. Thank you. And hopefully,
all right, guys.
Thank you. Thank you.
Have a good rest of the week.
Yes.
Yeah, you
Bye
too.
now.
Bye.